Hello, I'm Dr. Joachim-Friedrich Kapp from Aditxt, responsible for leading the development of a new drug for the treatment of autoimmune diseases. My fascination with immunology began during my medical studies. I became very interested in immunology because at the time, very little was understood of the immunological mechanisms. This led me to a postdoc at an institute for immunology to learn more about the immune system. My journey continued in the pharmaceutical industry, where I was involved in bringing the first treatment for multiple sclerosis to the market. Now, at Aditxt, I have the opportunity to spearhead research for a new drug for treating autoimmune diseases. Autoimmune conditions disrupt immune tolerance, where the immune system attacks its own body. The drug we are researching will restore immune tolerance by retraining the immune system. Every day, millions of normal cells in our body die and are replaced by new cells. This is what happens in the case of infections as well. The basis for the immune response, therefore, is a cell that died. That cell either carries a foreign antigen or it does not. In case it carries a foreign antigen, aggressive lymphocytes become activated, which direct themselves against the infected cells to eliminate them. If the dead cells do not carry foreign antigens, so-called regulatory lymphocytes are stimulated, which calm down the immune system to maintain the immune tolerance. Our drug consists of one component which is injected into the skin to induce a small amount of dying cells. As we learned, this is the primary condition and prerequisite for any reaction of the immune system. The second component of our drug, which is also injected into the skin together with the first component, is a slightly modified antigen from the antigen against which the immune system is directed in autoimmune disease. With these two major components of our drug, we successfully restore the lost immune tolerance, activating regulatory lymphocytes. In animal experiments, we have demonstrated compelling evidence that our drug restores tolerance in mice with type 1 diabetes and actively suppresses psoriatic reactions in animal models. These findings have positioned us to now transitioning from preclinical research to human trials. The German Health Authority knows our data and is expecting soon our submission to obtain approval to initiate clinical studies in patients. Starting this year, we plan to conduct a study in patients with type 1 diabetes and patients with psoriasis. We are optimistic that the promising results observed in animal models will translate into similar success in human patients, marking a new chapter in autoimmune therapy and hope for millions of patients worldwide.